Hello everybody. Today I'm going to be presenting to you part of my master's thesis, The Freshwater Fish Diversity of the Barra, Colorado Wildlife Refuge in Costa Rica, Traditional and Molecular Approaches to Biodiversity Investigation. Before we begin discussing the freshwater fish fauna in Barra, Colorado and in Costa Rica more broadly, we should first revisit and understand the value of biodiversity inventories. Diversity assessments can act as indicators of ecosystem health. By monitoring changes in species diversity, richness, or distribution, we can use this information to better understand how different things have impacted particular ecosystems and how ecosystem health is being impacted overall. And this becomes especially important in the presence of non-native species. For example, if we surveyed an area in 2015 and found 3 species present in that area, and returned to the area in 2020, finding only 2 of the 3 previously recorded species still occurring in the area, as well as the addition of 2 non-native species, this might provide us with information as to what particular areas we should be implementing additional conservation measures into, or how overall ecosystem health has been affected in that particular survey area. In order for us to conduct these biodiversity inventories or diversity assessments, we need to be able to identify the species present in an area of interest. And there are 2 general methodologies that are used for species identification, which I will discuss now. Morphology is the first that I would like to discuss. So morphology uses diagnostic morphological characteristics to identify specimens. However, it does require robust references and potentially experts, especially in areas or in particular groups where morphological overlap is a large issue and can make it more difficult for non-experts to utilize morphological characteristics to identify species. In addition, morphology does not address hidden or cryptic diversity, and this is common in highly diverse areas such as the Neotropics and in diverse taxa such as freshwater fishes. Because of this, DNA barcoding has been introduced as an established method for the identification of specimens utilizing barcoding genes, as it doesn't suffer from the same issue as morphology regarding morphological overlap in those diagnostic characteristics. For this project, we utilize the Cytochrome Oxidase Subunit 1 gene, as it is traditionally the first barcoding gene utilized for studying animals. In order for researchers to utilize DNA barcoding to identify specimens, they use public databases. So these public databases assign a species identity to a sequence by measuring the % similarity of the queried sequence to public data. Because of this, DNA barcoding also relies on high-quality reference libraries. However, previous studies have identified issues in this comparison step when utilizing public databases to identify sequences and consequently specimens. And this can actually prevent us from being able to use DNA barcoding as an effective measure for creating these diversity assessments and monitoring ecosystem health. So because of this, an integrative approach has been used previously, and this integrative approach uses both morphology and molecular identification techniques. However, it is less common, but by utilizing both, we can help to address some of the issues associated with morphology and DNA barcoding, and in the end, provide more robust identifications. If we move over to Costa Rica now, Costa Rica is a biologically important area with very high freshwater fish diversity, and this was the area we used for this project. Costa Rica sits within the Mesoamerican hotspot for biodiversity, and with its complex geologic history, diversity of habitat types, and climatic conditions, it contains about 5% of the world's global biodiversity. Because of this, Costa Rica relies on this biodiversity to help fuel its ecotourism sector, with ecotourism contributing the 2nd largest amount of the country's annual GDP after agriculture. With regards to the fish fauna, as I mentioned before, the fish fauna in Costa Rica is incredibly diverse. To provide a comparison to a potentially more well or better understood fish assemblage, we can take a look at Canada. So Costa Rica is currently estimated to hold around 250 species of fish and around 450 kilometers squared of total water area, with Canada holding around 225 species and over 1 million kilometers squared total water area. So this is quite a distinct difference in total water area, given especially in regards to the fact that these 2 assemblages are holding a similar number of species. So this helps to just illustrate how Costa Rica is incredibly diverse, particularly with regards to its freshwater fish fauna. For this project, we collected specimens in the Barra de Colorado Wildlife Refuge, which is located along the political boundary between Nicaragua and Costa Rica along the Caribbean slope, as indicated by the black box on the map here. As I mentioned before, Costa Rica relies on its diversity to help fuel the ecotourism sector, and because of this, the Costa Rican government has installed a large park and protected area system which covers about 25% of the national territory. And Barra de Colorado is one such wildlife refuge. So, Barra de Colorado was originally established as a link or a biodiversity corridor between Tortuguero National Park in the south and the Indo-Mayas Biological Reserve in the north and part of southern Nicaragua. These three areas together form the largest contiguous protected area of Atlantic lowland tropical wet rainforest in the world. While numerous studies have been done to assess the freshwater fish diversity in Costa Rica and in this wider Tortuguero region, of which the Barra de Colorado Wildlife Refuge is a part of, no formal studies have actually been done to assess the diversity of freshwater fishes in the Barra de Colorado Wildlife Refuge. This is problematic from a conservation standpoint, especially if this area is meant to serve as a biodiversity corridor between two other protected areas, and if we want to utilize this information to help us better understand ecosystem health. In addition, no studies have been done to assess the diversity of freshwater fishes in Costa Rica using molecular techniques. Because of this gap in knowledge, we had some questions we wanted to address. First, we wanted to try and determine what the current known diversity of freshwater fishes in Barra de Colorado is and how close is it to the actual diversity. Second, we wanted to determine if DNA barcoding is an effective tool for identifying freshwater fish species in Barra de Colorado. And finally, we wanted to assess if DNA barcoding could reveal morphologically cryptic diversity not currently understood using the morphological identification techniques. We conducted collecting expeditions during 2013, 2015, 2017, and 2019, and 10 sites were visited over these expeditions. During these expeditions, we used seine nets, dip nets, and gill nets to capture fish. Our collection sites were typically smaller and more shallow rivers and streams. These 4 collection efforts resulted in a total of 2,271 individual fish specimens captured. It is important to note that the sampling methods and the choice of collection sites likely impacted the species that could be found during this study, as the two were tended to favor smaller fishes. This likely resulted in the exclusion of larger species such as bull sharks or tropical gar, which are known to frequent our study area, however would be difficult to capture using the gear we collected and in the sites we visited. To identify the specimens we captured, all of our specimens were identified in the field using a 1993 dichotomous key for the region, and specimens were also then identified using morphology by curators at natural history museums where possible. In addition, we implemented DNA barcoding, and we were able to successfully sequence the Cytochrome Oxidase Subunit 1 gene for 240 of our specimens. In order to begin answering the questions we posed before, we had 3 objectives we wanted to address. First, we wanted to determine the species of freshwater fish present in Barra de Colorado using an integrative approach. First, we obtained morphology-based species identifications provided by experts when possible. Then, we obtained DNA barcode sequences from specimens when possible. Third, we compared our DNA barcodes not only to each other, but also to publicly available DNA barcodes. And by integrating these different data sources, we were able to determine species identity for 240 of our sequences. The results of this provided us with a better understanding of the diversity of freshwater fishes. Our integrative approach indicates the presence of 42 species from 35 genera and 17 families in the Barra de Colorado Wildlife Refuge to date. 6 additional species were also identified using high-quality photographs, which brings our total number of species observed in the area to 48. The 3 most commonly observed families were the characids, the cichlids, and the poecilids, and this is in line with other diversity assessments that have been done in other regions of Central America, with these 3 families being the most commonly observed. We also identified 2 non-native species, a hypostomus, a South American armored catfish, and a species of African cichlid. In addition, our study identified 7 species that were not known previously to occur in the area. Next, we wanted to test whether public DNA barcoding libraries for this study we focused on GenBank and BOLD would be an effective way to identify freshwater fish species in Barra de Colorado. First, we queried our barcode sequences for 240 specimens of the Barra, Colorado collection to the public databases BOLD and GenBank to identify them. We then compared the species identifications made by the public databases to the species identifications made using our integrative approach from the previous objective. From this, we found that the species identifications provided by the public databases matched the identifications provided using the integrative approach about 58% of the time. So for 141 of the 240 sequences, the species identification provided by both matched. The genus identification provided by the public databases matched the identification provided using the integrative approach about 99% of the time, so for around 238 of the 240 sequences. However, when using these public databases to identify our sequences, we encountered some difficulties, which we've classified into 3 broad categories. The first is misidentified sequences. So these are sequences in the databases that the species identity assigned to them may or may not be incorrect, but it did make it more difficult for us to utilize these public databases to identify our sequences. Second, the second largest category were species that were not represented in the databases. This made it difficult for us to assign a confident species identity using these public databases, since we weren't able to determine whether or not a specific sequence belonged to a species if there was no representative for it in the public databases. And finally, invalid taxonomy that was still being used for these reference sequences in the public libraries made it difficult for us to utilize these to identify our sequences. Based on the issues that we identified when using the public DNA barcoding libraries to identify our sequences, we wanted to build a curated reference library for the freshwater fishes in Barra, Colorado. First, we created a list of species that were likely to be present in our study area based on literature. Then we added barcoded sequences from the Barra, Colorado collection that have been associated with a physical specimen. This was just to keep it in line with the traditional rules and regulations associated with building a library. Then we added additional representative barcode sequences from the public databases GenBank and BOLD. However, because of the issues with using public data, we wanted to only utilize high quality public data, so we created some criteria for the critical evaluation of these public databases to ensure that we were only including high quality public data in our library. The results of this, when we generated a regional species list using the previous diversity assessments, but we focused on the wider Tortuguera region of which the Barra, Colorado Wildlife Refuge is part of, because there haven't been any studies done in Barra, Colorado. And this wider regional library contained 162 species from 105 genera and 43 families. Our current curated barcode reference library contains 137 species. 38 of these species are represented by sequences from the Barra, Colorado collection, and 99 of these species are represented by high quality, critically evaluated public data sequences. Of our 162 species on the regional list, 25 are missing from the library because there is no representative sequence for these species in the public databases. And this includes 3 species listed on the IUCN list, 1 vulnerable, 1 endangered, and 1 critically endangered. We are currently working to transform this curated barcode library into an accessible tool for conservation managers and researchers. This is the first study that's been done to assess the freshwater fish diversity in Barra, Colorado. It is also the first study that's been done to address the freshwater fish diversity in Costa Rica using molecular techniques. We were also able to detect 2 non-native species in the region, and the tools resulting from this project can be used to aid in biomonitoring efforts, particularly when they're done by non-experts, as our tools do not suffer from the same issues typically associated with morphology or public DNA barcoding libraries. And finally, the biomonitoring information can be used to inform conservation managers and policymakers, and at least that is our hope in the future, that we will be able to provide additional and better tools for conservation managers and policymakers. We would like to thank Dr. Lopez Fernandez and Arturo Angulo for their identifications of the specimens. We would also like to thank the Canadian Organization for Tropical Education and Rainforest Conservation for their help in facilitating the fieldwork. We would also like to thank members of the Lovejoy Lab for their continued support. And finally, we would like to thank the University of Toronto Scarborough for their support in this project.